Protein purification by immobilized metal affinity chromatography. The target for purification is the HfxI recombinant protein sourced from a salt marsh environment in Canada. HfxI is trimeric and is of particular interest due to a novel protein fold. In order to isolate our target protein, it must be extracted from the complex mixture of the cell upon disruption of the cell wall. Methods of separation rely on targeting the unique physicochemical properties of the protein. Immobilized metal affinity chromatography was undertaken using a chelated metal species attached to a solid support. The metal ion in the nickel Nt resin used has a high affinity for polyhis tags. As such, recombinant HfxI associates with the column while other non-specific protein species can be washed out. This association is disrupted in order to retrieve our target by an increased concentration of imidazole, which outcompetes the polyhis tag for association with the immobilized nickel ions. In one chromatographic step, the protein of interest can be isolated from most of the cell contents. However, this technique is vulnerable to non-specific binding of proteins with adjacent Is residues. In order to end up with as much purified protein as possible, growth conditions of the expression system must be optimal. Our E. coli expression system was doubly protected by the chloramphenicol gene in the PLYSS plasmid and the ampicillin resistance gene present in the plasmid encoding the HfxI protein. The cells are provided with the nutrients and conditions ideal for growth. 5052 media contains a mixture of sugars the cell will sequentially use up until lactose induces the expression of our protein. The culture underwent centrifugation and the cell pellet was resuspended in binding solution E. The cell wall of E. coli must be disrupted in order to access the targeted protein. This is achieved by the enzymes lysozyme and DNase1. These enzymes hydrolyze bonds and break apart these unwanted biomolecules. Physical disruption of the cell takes place by exposing the sample to large temperature changes, by snap freezing and then thawing with a water bath for 20 minutes. This is followed by sonication to further lyse the cells. There is a balance to be kept between disrupting the cell and maintaining the integrity of our target protein. Protease inhibitor was added to disrupt the action of proteases in the cell that could target our protein. Following centrifugation, the supernatant was applied to a 0.4 micron filter to remove large contaminants. Ethanol was spun through the Imac column containing nickel Nt resin. The column was then equilibrated in binding solution E. Successive column volumes of filtrate, wash solution B and elution solution C were spun through to HfxI, wash non-specific proteins, and elute Is tagged protein, respectively. The concentration of imidazole increases to first disturb weak associations of contaminants, and then elute out HfxI. Visible in the SDS page gel are the successive levels of purification achieved from total cell lysate to filtered and then Imac purified HfxI. Immobilized metal affinity chromatography separates targeted tagged proteins from the majority of cellular proteins in one time and cost-effective step. While Imac has its disadvantages, it is a highly favored chromatographic technique that isolated the recombinant HfxI successfully.